This is the first of 5 segments on contingency tables, a subject also known as cross-tabulation. This is a very important subject, and it's one that a lot of people get wrong. So I hope that after viewing these 5 segments, you'll really understand contingency tables. Here's an example of a contingency table. This is a standard textbook example that is used in a number of places. A contingency table has a column condition. In this case, it's alcohol consumption. And it has a row condition. In this case, absence or presence. And then it has counts of events in every cell in the table. So for example, this one is a study where more than 30,000 pregnant women were asked about their alcohol consumption, and on that basis they were assigned to one of these 5 columns in the way that you can see here. And then what was tracked was whether their infants, when born, had any malformations, absent the good case, of course, present the sad case. And the question here is, of course, is alcohol implicated in malformation? Now, the question of whether alcohol consumption causes malformation is a causality question which contingency tables don't directly answer. A contingency table is capable of showing statistically that there's an association between a row variable and a column variable. And on the basis of that association, one may go on either to impute causality or to do additional studies to figure out which direction, for example, the causality goes. This kind of data is very often used in setting public policy, so it's important that we be able to assess its statistical significance. Let's just look at what the numbers are. If we go all the way up here to the pregnant women who had more than 6 or more drinks a day, imagine that, we see that there was 1 malformation and 37 in which the babies were normal. So that's 1 part in 37. Now if we come all the way down here to 0, well, let's do the numbers roughly. We have 50 out of 17,000. So that's about 1 out of 340, which is certainly a much smaller percentage than 1 out of 37. In fact, here in this next column we have 1 out of 126, and that's also rather larger than 1 out of 340. But if you're worried about statistical significance because the counts here are so small, 5, 1, 1, then you're worried about exactly the right question. And that's what we want to learn how to deal with. So let's take a simpler example, a 2 by 2 contingency table. We're going to go back to the human genome, where, if you'll recall, genes have 1 or more exons. And you'll also recall that genes are made of nucleotides A, C, G, T. And some genes are more rich in A's and T's than they are in C's and G's. There's a whole story behind that. So we might ask a question like, is a gene more likely to be single exon if it's AT rich? In this case, the row variables I'm going to take as, does the gene have 1 exon or does it have more than 1 exon? And the column variables, well, AT richness is measured as what fraction of the nucleotides are either A or T. And you'll notice here I've just decided that I'm going to define richness as being greater than 0.6 for that fraction, and non-richness as being less than 0.4. In other words, there's no necessity that either the row conditions or the column conditions be exhaustive and mutually exclusive. It simply matters that you can figure out how many events are in the intersections of the row and column conditions. So in MATLAB, one convenient way to do this is, given a vector of data, so here's a vector of cells G, one for each gene, and each gene has some properties like NE, the number of exons, or ATF, the fraction of AT nucleotides. And here I've set this up in MATLAB as a matrix of logical conditions. So each row in this matrix is 1 gene, and it has two columns, each of which is either true or false, 1 or 0, and in this case the entry is true if the gene's number of exons is equal to 1, and the corresponding entry here in the same row is if the gene's number of exons is greater than 1. So that's the row condition. The column condition is G a t fraction less than 0.4, true or false, gene a t fraction greater than 0.6, true or false. And when you've organized the data this way, it's easy to define a function contingency table that just goes through and counts it. You could do this by hand if the number of counts were smaller than shown here. And these are the counts that come out of the human genome for this contingency table. You can see that there are fewer genes that are AT rich, that is to say, this column, where the column condition AT greater than 0.6, there are fewer than AT poor, and you'll see that there are more genes that have more than one exon, that would be the second row, than the first row. It's often useful to consider the marginals, the sums of a contingency table. So in this case, I'm going to sum along the first dimension, that is along the row dimension, and then for each column I get a total, the column marginal. You can see that this is the sum of these two, this is the sum of these two. And now if I divide each entry by its column marginal, I get a set of probabilities where the probabilities in each column will add up to 1. So we can look at this and we can see that the AT-rich genes, which is this column, have slightly fewer single exon genes, 14.7%, compared with this column, which has 15.1% for the AT-poor genes. Well, that is a difference, but it's such a small difference that we rather suspect that it's not statistically significant. Or maybe it is, because we really have a lot of data here, and maybe we can detect that small difference, and it's small but significant. That's what we need to find out. Now it turns out that this contingency table function here doesn't actually exist in MATLAB. MATLAB has a contingency table function, but it doesn't put out the output in a format that I like, so I wrote my own here, and this is one of these MATLAB things that you'll have to decode from the inside out, because it has repmats, repeats of rows or columns of matrices, and sums and squeeze and permute, and I'll leave it as an exercise to you to figure out how this works if you're a MATLAB coder. We already talked about the Pearson statistic for data which comes in the form of counts. We previously talked about this in the case of histograms that come in the form of counts, but even then I forward bookmarked that we were going to do this test also on contingency tables. So let's now look at it. We're going to need a bit of notation. A very convenient notation in contingency tables is the so-called dot notation. That means that if I have the matrix of counts, that here I'm going to call Nij, where I labels the rows and J labels the columns, and I want a marginal, I can denote that marginal by just replacing one of the indices by a dot. So Ni dot is the ith row and the sum of all the columns, and here's the formula for it here. Similarly, N dot J is the sum of all the rows in the jth column. There it is. And finally, of course, there's the total number of counts in the whole table, which is either the sum on I of Ni dot or the sum on J of Nj dot. So I could write this as N dot dot, but I'll just write it as capital N. Now, what's the null hypothesis on a contingency table? That's always clear. The null hypothesis is that the row variables and the column variables have no particular association. In other words, which row an event falls in is an independent event from which column it falls in. And in that case, if you think about it, if you take the total number of events in row I independent of which column it's in, so I'm doing ni dot, and you divide by the total number of events, then this is the row probability, or at least an estimate of it over the data. And that row probability ought to be repeated separately for each column, so that each individual nij should be that same fraction of n dot j. If you now multiply this through by n dot j, you'll see that what we have here is the expected value that we expect in a cell nij can be formed from just these marginals and the total number of events. In other words, it's the row marginal ni dot times the column marginal n dot j divided by the total number of events. So that's the model of the null hypothesis for a contingency table. And now the Pearson statistic is just the same as we've seen before. Before we used the mnemonic for it that it was observed minus expected squared divided by expected. And we saw that that was a chi-square-like statistic. There were some important footnotes on that because it's not exactly chi-square distributed for small counts, you should recall. In this case, the number of counts is very large, so we can ask, are the conditions for this to be approximately chi-square distributed valid? Yes, because the number of counts in all the bins is very large. Now what if the number of counts in one or more of these bins was small? What if it was 1 or 5 or 3? Well, then this statistic would not actually be chi-square distributed. Furthermore, we couldn't use that trick that we learned about in fixing the moments of it in a previous segment, because that trick only worked when we had a large number of bins. So the interesting thing about contingency tables that we're going to have to come back to is that it's quite possible to end up with both a small number of bins, that is to say entries in the table, like 4, and a small number of counts in one or more of the entries. So that's a case that we're going to have to learn how to treat. But not yet. Here we have large numbers of counts, and we can just do the chi-square test. So here I'm computing the expectations of the table, the null hypothesis table, and this, if you peek through the MATLAB, you'll see is just this formula up here, and that gives count numbers here. They look a little funny because MATLAB wrote it as 10 to the 4th times these things, but you can see this one is 2,372 and so forth. Then the chi-square formula right here becomes this in MATLAB. The chi-square value is 0.43. And what do I compare that against? Well, I compare it, of course, against the chi-square cumulative distribution function because I'm doing a p-value test, a tail test. MATLAB has that function, but I have to know the number of degrees of freedom. So that's a little bit tricky here. Let's think about it. There were 4 bins, but we fixed the marginals, both the row marginals and the column marginals in our model. We fixed them up here when we computed the expected values. So we have to subtract 2 degrees of freedom here for the row marginals and 2 degrees of freedom here for the column marginals. But wait, that's overdoing it because row and column marginals both add up to the same number. They add up to the total number of events here. So we overcounted and we add back the one here. And this, I didn't write it down, but this is a general formula. The number of degrees of freedom in a contingency table is the total number of cells minus the number of rows minus the number of columns, and then don't forget the plus 1 here. So I put that into the cumulative distribution function, and I get a tail probability of 0.49. Well, that's positively embarrassing. After all, 0.5 is the least significant possible tail probability that you can get for a two-tail test. So we see that, in fact, there is no evidence of an association between a gene being single exon and a gene being CG rich, even when you do the test very sensitively by using all of the data of all of the genes in the human genome. Now let's get back to this question about when the number of counts are small, because we're going to see that some subtle issues show up. So in the remainder of this segment, I'm going to just define some notation, and then we're going to review a couple of probability distributions that are going to come into it. And then in the next segment, we'll really leap right into these issues. So the setup, to be a little more detailed, is that the columns are often called conditions, and in a typical biomedical application, the conditions might be, is the patient healthy, the control group, or is the patient sick, in a group of patients who have reported to a hospital with some set of symptoms. And the factors, which are generally taken to be things that are more under our control, that's just a convention of which you put in the columns and which you put in the rows. The factors here might be, was the patient vaccinated or not? Maybe vaccinated is F1 and non-vaccinated is F0. And then here we have in the cells the number of counts in each of these four possibilities. From those counts, we have the marginals, the row marginals, n0 dot, n1 dot, and the column marginals, n dot 0, n dot 1, and the total number of events. Here I've written it as n dot dot. So the null hypothesis is conditions and factors are unrelated. And we want to do an honest, accurate p-value test. So how do we do that? We, number one, invent a statistic that measures deviation from the null hypothesis. We're very firmly in the frequentist camp here when we're doing contingency tables, so we're allowed to invent any statistic that we want. But there are some statistics that are more sensitive than others, and there are some that have been honored by history and have become a common language of contingency tables more than others, so we'll generally use statistics that are well known. Then we'll compute that statistic for our data. We've already done that when we used the chi-square statistic in the previous slide and computed the value of chi-squared for the data in the particular contingency table that is our data. Then step 3, and this is where the subtlety comes in, we need to find the distribution of our statistic under the null hypothesis over the population of all possible contingency tables that we might have seen but didn't see, because then that's the population where we'll see where is our real data in the tail and what's the value of the tail probability. Now number 3 here is the hard part, and we'll see is the subtle part, because this is not an obvious question. What is the population of contingency tables? Is it some infinite set of all possible integer numbers that we can put in all possible 4 bins of that? Well, that's an odd distribution, and as the numbers get very large, that distribution just goes off to being of infinite size. So that's not going to be the right one. In fact, we're going to see that from what I've told you so far, there is no one right answer to this, because it depends not just on the numbers in the contingency table, it depends actually on the experimental protocol under which that data was gathered. And that's the subtle point in contingency tables that we're going to come back to in the next segment. Sometimes this dependence is only slight, and so what I'm talking about here is a mere academic footnote. But sometimes that dependence can be important, so it's important that we understand it. Now let's review in the end of this segment a couple of probability distributions. Here's the hypergeometric distribution. It answers a question like this. There's a car race with 3 teams, one driving Ferraris, the second driving Renaults, and the third driving Hondas. And each team has entered 6 cars into the race. We want to ask, what is the probability of the car race finishing with 2 Ferraris, 2 Renaults, and 1 Honda constituting the top 5 finishers? Now, of course, we want to ask this under the null hypothesis. And the null hypothesis here is that each race car has an equal probability of finishing in the top 5. So let's calculate that. Well, that's an easy probability problem. We just need a bunch of these choose functions, which we know are binomial coefficients. So the total number of ways we could have 2 Ferraris finish in the top 5 would be the number of Ferraris, that's 6, choose 2. And the total number of ways we could have 2 Renaults is also 6, choose 2. And that's, of course, independent of the Ferraris. And the number of ways we could get 1 Honda is 6, choose 1. So the numerator is the product of these 3 binomial coefficients over here. And the denominator is the total number of distinguishable ways of having a top 5 cars. So that's the sum of total number of Ferraris, Renaults, and Hondas in the race divided by total number that we're assuming finish in the race. That's going to come out 5. And so you see that's 18, choose 5. So the answer to this particular little probability problem is about 16%, as you see here. Now what makes this a hypergeometric distribution is this property that you have a product of chooses in the numerator, and then a denominator which is the sum of the top rows of the numerator and the sum of the bottom rows of the denominator. Hypergeometric distributions sometimes appear in slightly different problems. So here's a slightly different case, but we'll see that it also gives a hypergeometric distribution. Suppose we have capital N genes, might be 30,000 genes, and we have discovered that little m of them are associated with a particular disease, disease 1, and little n of them are associated with a particular disease, disease 2. R genes are in common between these two sets, and we want to know what's the probability of that happening by chance. So the picture is shown here. The total number of genes is n. We have m associated with disease 1, n associated with disease 2, and here in the center r in common between the diseases. You can see why this is an interesting problem, because it might be that these R genes are simply chance associations, or it might be that what we've discovered is a relation between the two diseases, that they share a common set of genes that causally are involved in causing both diseases. So how do we calculate this? Okay, the denominator is just going to be how many different ways could we have randomly picked the M genes for one disease and the N genes for a second disease. So that's capital N choose little m and capital N choose little n. And it's the product of the two because those chooses can be done independently. Now what about the numerator? The numerator here is, well, let's start with m. How many different ways can we pick the set of m? That's n choose m over here. Now, of those m, r of them are in the overlap, and we have to figure out how many different ways we could choose this overlap region. We have to choose them out of little m possibilities, so that's m choose r. Then we have to choose not the set n, because we've already chose r of them, but the rest, this little moon-shaped region in blue of this set. And so you can see that that set is chosen from a number of possibilities, capital N minus m, and the number that we're going to choose is little n minus r. Okay, so that's the reasoning through what the formula is. Notice that there's a term in common in the numerator and denominator, here and here. And if you cancel those out, you get the result over here. And you can see that this is a hypergeometric distribution. Why? By definition, because the sum of these things up here, m plus n minus m, gives n, which is the term in the denominator, and r plus n minus r gives n, which is the term in the denominator. So in this kind of a problem, we also get a hypergeometric distribution. Now written like this, it doesn't show that it's symmetric between little m and little n. It has to be because you can see this picture is symmetric. But if you write out all of these choose functions, these binomial coefficients, in factorials in the usual way, and then cancel various of the pieces, you can see that this is the formula for the hypergeometric probability distribution, and it is symmetric on little m and little n. And we're going to write this in the case where there are simply two products in the numerator. Up here was a case where we had three products in the numerator, but in the two case, we'll write this as hypergeometric probability of finding R overlaps out of n total genes where m is in one set and n is in the other set. Now let's review the multinomial distribution. The idea here is that on each independent and identically distributed trie of something, there are exactly one of capital K outcomes. And the probabilities of each of those outcomes is some set of probabilities, P1 through Pk, which add up, of course, to 1, because they are exhaustive and mutually exclusive. So now we want to ask, if we are going to do n tries, what's the probability of seeing the exact set of outcomes, little n1 of the first type, little n2 of the second type, and so on up to little n sub k of the kth type? Now, we did capital N tries, so these number of events have to add up to capital N. And that is the multinomial probability distribution, which we'll write as the probability of seeing this vector of numbers of counts if we do n tries of something that has this vector of probabilities of each of the alternatives. So this probability is, I'm sure I'm reviewing material that you've studied before, one place or another, this is the probability of any one particular outcome, and a particular outcome would say the first event comes out maybe in bin 6 here, so that would be somewhere here in the middle here, a factor of p sub 6, and the second count comes out in maybe the first bin, so that would be a factor of p sub 1, and in the end, when we've calculated them all up, we'll see that we'll have a number of factors of p1 that's exactly equal to its number of events, factors of p2 equal to its number of events, and so forth. But the probability of seeing this number of n's is not just the probability of seeing one particular instantiation. We have to multiply this by the number of equivalent arrangements, and that's given by this combination of factorials. Total number of events factorials divided by the product of each of the individual number of events factorial. Well, again, this is a formula that you've probably seen derived before, but let me indicate here where it comes from. Let's do the special case where there are exactly 26 events. And suppose we measure it and it comes out that we see 5 events in the first bin. So just to space my line, I've written 1, 2, 3, 4, 5 here, and 3 events of the second type, 1, 2, 3, and 8, 1, 2, 7, and so forth. Actually, not and so forth, because these add up to 26, the total number of events. So the total number of ways I can assign the events to this pattern is, I'm going to think of my events as labeled A through Z. That's why I picked n equals 26, of course. And one way of writing out the 26 letters of the alphabet is in order, but there are, of course, 26 factorial or n factorial arrangements of the alphabet, and each of them is therefore an assignment into these sets of bins, and they're n factorial, the numerator, possible sets of assignments. But not all of those are distinguishable, because any permutation of these 5 events is indistinguishable, therefore divide by n1 factorial. Any permutation of these 3 events is indistinguishable, therefore divide by n2 factorial, and so on. That's all we need to know about the hypergeometric and multinomial distributions, and we'll use them in succeeding segments.